And now we're going to talk about some growth requirements. It's going to be fairly short and sweet. If I can do it, I'm not going to like push it through, but it would be nice to just wrap it up. So let's target the most complicated part first. Okay, it's not really complicated, honestly. So first is going to be oxygen requirements. You need to know 5 different categories of microorganisms based on the oxygen requirements. First is super simple obligate aerobes. They grow on top of the tube. That makes sense? I'm going to make it a little prettier. Okay, so they grow on top of the tube. They need oxygen. Like need oxygen. What kind of metabolism would they use? What aerobic respiration, anaerobic respiration, fermentation, what do you think? Aerobic respiration. Okay. Then you have obligate anaerobes. So these fellows will grow at the bottom of the tube, right? Oxygen is toxic for them. What kind of metabolism they would use? Most likely for you. I understand the thinking. We'll get to why it's most likely fermentation. You will see it's really elegant idea here. Okay. Now I'm gonna get progressively more complex ideas. Okay. So next one will be aerotolerant anaerobes. Okay. Throughout the entire tube, no preference for oxygen. So oxygen is not needed. Is it toxic? No. So now, Julianne, what can be? Anaerobic respiration or fermentation. Bless you. Microaerophiles. Grow at the very exact concentration of oxygen. So exact oxygen concentration is needed. Does that make sense? These are called, usually it's pretty low, like 1% oxygen. These microorganisms are called Goldilocks. Okay? Because of that, story. Okay? What kind of metabolism? If they need oxygen. I'm not showing. What kind of metabolism? Aerobic respiration, yes. And finally, facultative anaerobes. So this is interesting. For instance, E. coli is facultative anaerobe. Okay, so look, it can grow with or without oxygen, you see that? But, so, can it grow without oxygen? Yes or no? Yes. But it grows better with oxygen. So we can say that oxygen is preferred. E. coli, Staphylococcus aureus, they are facultative anaerobes. You grow them in anaerobic conditions, fine. You grow them in aerobic conditions, fine. So they can do both aerobic respiration and fermentation. And think of it this way: they can easily switch, right? Like you do. They just at this end of glycolysis, they kind of decide. Oxygen is present. Oh, we're going to respire. Oxygen is absent. We're going to ferment. In reality, though, in the presence of oxygen, they're going to do both. Does that make sense? They're going to ferment some, and they're going to respire. They're going to run the full oxidative respiration gig. We're good so far? Now, addressing this, why mostly fermentation? So there are 3 enzymes that are essential to neutralize reactive oxygen species. So I'm gonna just hang on with me. Neutralizing reactive oxygen species. We have 2 species that are most important. It's superoxide anion here on top, O2 minus, and hydrogen peroxide. So superoxide anion can be neutralized by superoxide dismutase. Okay? And hydrogen peroxide can be neutralized by peroxidase and catalase. Okay? Now, superoxide anion is formed almost everywhere where oxygen is present. Does that make sense? That makes sense. Hydrogen peroxide is mostly formed during the respiratory processes. Now look at this. I'm going to put this. So enzymes. Obligate aerobes. Are they exposed to oxygen? Yes. Do they respire? Yes. So these guys usually have all 3 enzymes. 1, 2, 3. You with me? Obligate anaerobes. Why are they obligate anaerobes? Because they don't have any of the enzymes. So Julian, to our conversation, if they would do anaerobic respiration, check this out, they would do anaerobic respiration, they would produce hydrogen peroxide. They would need to neutralize them. They don't have it. Okay? Now, air-tolerant anaerobes, peroxide, usually catalase. Sorry, not catalase, superoxide dismutase. Okay? So like, they can deal with, the most dangerous one here. They can deal with the most dangerous one. Now, hydrogen peroxide, they're okay. They can tough it out. Microaerophiles usually have all. Now, facultative anaerobes, SOD, catalase, maybe peroxidase. Okay? Now, before we move on, before I ask you, do you have any questions, I want to kind of highlight what you need to know. You have to have a pretty good understanding of like the mode of growth, where in the tube they grow. Is that clear? What is the role of oxygen? Do they need it? Is it toxic for them? Stuff like that, okay? What kind of metabolism they use. I'm not going to ask about the presence of enzymes, but you certainly must know what these enzymes do. So if I ask you which enzyme neutralizes hydrogen peroxide, this or this, please don't invent new ones. Okay? Go ahead.
Can you just repeat that?
Mm-hmm.
I was just a little confused.
Of course. So, again, mode of growth, these little tubes, I can ask, you know, I'm going to give you, among others, a description of a microorganism saying, this microbe grows blah, blah, blah at the top of the tube. What is it? Exactly. Things like that. Make sense? Role of oxygen. Does it need it? Does it not? Is it toxic? Is it not? Maybe it doesn't need it, but oxygen is okay. That's going to be your air tolerance. Does that make sense? This kind of stuff. Type of metabolism. And what these three enzymes do. Good? Again, do not invent your own enzymes. That happened before. Okay. Make sense? I think I'm being pretty straightforward. pH requirements, this is ridiculously simple. Okay? Acidophiles, alkaliphiles, neutrophiles. Everybody remembers their pH? Okay. So, less than 7, more than 7, around 7. Most human pathogens are what? What do you think? Most human pathogens? Neutrophiles. Neutrophiles. Because actually acid is very protective. Like acidic environment in the stomach, acidic environment in the vagina. There are some acidophiles in the vagina, but that what protects vaginal mucosa from infection. Does that make sense? Most human pathogens are neutrophiles. Temperature. So first category. Psychrophiles. Okay? Grow in the fridge. You probably noticed that in the notes I provide you with exact optimum growth requirements, like it says, like below 0 degrees Celsius. I want it to be more qualitative than quantitative. Okay, so this will actively grow in the cold and will not grow at the room temperature. Am I clear? Psychotrophs. Slowly grow in the fridge. Normal growth at room temperature. So this is the problem. These microorganisms can spoil food. You put the food in the fridge, but they keep on growing. Does that make sense? Very, like Listeria monocytogenes is a famous psychotroph. Mesophiles. Room temperature, 37 Celsius. Okay, human pathogens. Most human pathogens are here. You good? Good? Thermophiles. Scalding water. Not yet boiling water, but pretty hot. Okay, so we're talking about 50, 60 degrees Celsius optimum. Am I making sense? And extreme thermophiles. Boiling water. This is why, look, this is why I want to make it qualitative but not quantitative. Because it's much easier to remember, you know, like this microorganism can grow in a boiling water pretty nicely, than to remember 100 degrees Celsius. Does that make sense? So I'm going to be asking you questions qualitatively. Like, if I ask you, something that grows pretty well in the fridge but not at room temperature, tell me that's a psychrophile. Am I clear?
Is this what you meant by saying that this is not Thursday's exam, but Tuesday's exam? It's going to be a lot of memorization. This is the memorization?
This is the memorization. This is like a lot of memorization. Yeah. There's not going to be... Basically, biogeochemical cycles is more of a concept, I would say. This is just, those are the terms, those are the terms, those are the terms. There's going to be some concepts in antibiotics then. Osmolality requirements. Now you remember, osmolality is the concentration of anything in the solution, right? So microorganisms that can grow in the presence of high concentration of solutes called osmophiles. Am I clear? Any solute, sugar, salt, protein, whatever it is. Am I clear? Does that make sense? Now, within osmophiles, we identify a special category of microorganisms called halophiles. Specifically, high concentration of salt. Specifically, salt. This is important because salt, we use it for conservation purposes. It makes sense? Good? Makes sense? Okay. So, all halophiles are osmophiles, but not all osmophiles are halophiles. Am I clear? Okay. Again, do not invent your own terminology. Last few kind of garden variety terms here. Xerophiles, dry environment, okay. Barophiles, high pressure, basically bottom of the ocean. There are microorganisms there. And extremophiles. I should say polyextremophiles. All kinds of stuff. My favorite, Deinococcus radiodurans, can grow in the presence of ionizing radiation, high temperature, and boiling water, and acidity, and all four at the same time. And they're going to be just fine. Does that make sense? Now, those questions usually, no problem, okay? And I promise you, and you probably noticed it, that I'm not going to put you in the awkward position, for instance, if I tell you, microorganism that grows at 90 degrees Celsius and put thermophiles and extreme thermophiles in the same list. It's impossible to distinguish. It's like two gray areas. Does that make sense? Okay. Last bit to kind of wrap up the first unit, and it's great. We're doing phenomenal. It's going to take 5 minutes. Microorganism is an example of microaerophile. Campylobacter jejuni. So this is a microaerophilic and on top of that it's also capnophilic. So capnophilic means it requires CO2. Okay? So very specific concentration of oxygen, elevated concentration of CO2. This is what Campylobacter jejuni requires. What does it cause? It causes gastroenteritis. Okay? Garden variety gastroenteritis with diarrhea, vomiting, abdominal cramps. Does that make sense? In the rare cases It can cause Guillain-Barré syndrome, which is autoimmune condition. Usually it is transient. There's a treatment, people receive intravenous immunoglobulins, and usually Guillain-Barré syndrome is successfully treated. So far, so good? Make sense? Now, why we pay so much attention? Because it's a zoonotic disease, and usually people receive it from underprocessed or unprocessed milk, poultry, or eggs. Very much like salmonella. Does that make sense? Telling it's gram-negative. Does that make sense to you? So, gram-negative, microaerophilic, capnophilic microorganism, gastroenteritis can progress to GBS. Transmission through the food that is sourced from animals. This is why we pasteurize milk. This is why we boil eggs. And this is why we clean our kitchen after we work with chicken. Okay? Now, it's interesting that we have species of Campylobacter other than jejuni in our mouth. And it's been observed, not shown, observed, that there is an association between Campylobacter in the mouth and inflammatory bowel disease. But there is no association, interestingly enough, between this one and IBD. So that's interesting. Like this one is an acute disease, you see? Like you eat it, you get gastroenteritis, you suffer for a week maybe, take antibiotics, recover. You have Campylobacter in your mouth, you maybe they, we don't know if they cause it, but maybe they do. Does that make sense? It kind of shows you the variety of effects that microorganisms within the same genus can have in a human. Questions? Okay, awesome.